Assalamu alaikum. This is me, Atiq Ahmad. Dear students, today we are going to study two organelles, the Golgi apparatus and the mitochondria. These two are important organelles found in the eukaryotic cells. We will start from the Golgi apparatus. Golgi apparatus is basically a set of flattened sacs. They are a set, a combination of several flattened, means flat sacs, which are called cisternae, which are stacked together, which are combined together to form a stack. In local language, we call it dere, stack. So Golgi apparatus is basically a set of flattened sacs. Those sacs are called cisternae, which are stacked together. It was discovered by Camillo Golgi, a scientist. So it was named as Golgi apparatus. Diagrammatically, this is Golgi apparatus. This is Golgi apparatus. These are the sacs. One sac, two sac, three sac. This is another sac. This is another sac. This is another sac. These are called cisternae. These flattened sacs, they are combined together to form a stack. We will move towards the teaching points. The function of Golgi apparatus is to store the secretions, proteins, then convert them into finished products, and then pack them into small rounded sacs called Golgi vesicles. Now, what happens basically that the ribosomes is basically the protein synthetic machinery. Ribosomes are either freely found in the cytoplasm or they are attached to the rough endoplasmic reticulum. So through the ribosome, proteins are formed. If those proteins are in the final shape, if that is the finished product, there is then no need of further processing. There is no need to convert them into finished product. So the proteins which are prepared on the ribosome, they will be directly utilized by the cells or they will be transported outside through the endoplasmic reticulum. But those proteins which are formed on the ribosomes and they are not in the form of finished product, so first they need to be processed. So that processing is basically performed by the Golgi apparatus. Golgi apparatus is just like a company. Here raw materials are brought, raw materials are first stored and then they are converted into finished products and then they are packed into small packets, small rounded sacs called Golgi vesicles, and then those small sacs, small pockets are then transported outside the cell or inside the cell wherever they are required. So the function of Golgi apparatus is to store the secretions, proteins, those proteins which are in the form of raw materials and they must be first processed to convert them into the finished products. So one of the function of the Golgi apparatus to store those raw materials, those secretions, those proteins. Then they will process them to convert them into finished products and then they will be packed into small rounded sacs called Golgi vesicles. Then these Golgi vesicles, if required inside the cell, they will be transported inside the cells. If they are required outside the cell, so they will be transported to the exterior. Exterior means outside in the form of secretions. Secretion in biology, anything which is released, which is secreted, is called secretion. Now another organelle we are going to study, that is the mitochondria. Mitochondria are only found in eukaryotic cells. They are not found in the prokaryotic cells. Prokaryotic cells means bacteria and cyanobacteria. So they are found only in the eukaryotic cells in animals, plants and fungi and some protist cells. And they are the site of energy production because the process of respiration takes place inside the mitochondria, so they are the site of energy production. Mitochondria are absent in prokaryotes. So how they produce the energy? So they have alternative methods. they use the process of anaerobic respiration, which is the process of respiration without oxygen. Similarly, mesosomes are present in bacteria and cyanobacteria, which are enfoldings of the cell membrane. So in prokaryotes, means bacteria and cyanobacteria, there are no mitochondria. So the alternative methods they have, that they have anaerobic respiration, they produce their energy without the use of oxygen, or they have mesosomes enfolding of the cell membranes, which are utilized for the production of energy. Now we will discuss in detail the eukaryotic mitochondria. Mitochondria are covered by the two membranes. This is a double membrane organelle which consists of two membranes. The outer membrane is smooth while the inner membrane is folded inwards, which give rise to small extensions called cristae. Now these cristae increase the surface area on which the respiration process takes place. Diagrammatically, it is here. So this is the outer membrane, this one, this is the outer membrane. This is the inner membrane, the second membrane which folds inside. These are the folds of the inner membranes. These folds, these and these folds are called cristae. And in between there is a matrix. This is called mitochondrial matrix. This one, light bluish one. This is mitochondrial matrix. So mitochondria is a double membrane organelle which consists of two membranes. It is covered by two membranes. The outer one is smooth and the inner one makes infolding inside. Those foldings are called cristae. Those are called cristae. Cristae increase the surface area on which the respiration process takes place. Mitochondria is known as powerhouse of the cell because they perform the process of respiration to produce energy in the form of ATP. Now the carbohydrates in the form of glucose, they are broken down inside the mitochondria. So as a result, they are converted to H2O plus CO2 and the stored energy is released in the form of ATP. That ATP is an energy rich molecule and it supplies energy for life activities of an organism. So this is all about today's lecture. Next time inshallah we will meet with another fresh lecture. See you next time. Take care. Allah Hafiz.